Hello and welcome to today's GeneWiz Week webinar, Rewritable Multi-Event Analog Recording in Bacteria and Mammalian Cells. I'm Jay Shah, Product Marketing Associate for Gene Synthesis at GeneWiz, and I will moderate today's webinar. I'm very pleased to introduce today's presenter, Dr. Weixin Tang. Dr. Weixin Tang obtained a bachelor's degree in chemistry and biology from Tsinghua University in China. After moving to the United States, she completed her PhD with Professor Wilfred van der Donk at the Department of Chemistry at the University of Illinois. She is now an HHMI Fellow at the Jane Coffin Child's Memorial Fund for medical research for Professor David Liu's lab at the Broad Institute. If you have any questions for Dr. Tang during the webinar, please enter them into the Q&A section, and we will try to address them as many as possible at the end of the session. With that, please welcome Dr. Weixin Tang.
All right. Before I start, I want to first thank GeneWiz, especially Jay and Lily, for inviting me to do this webinar. I take this opportunity as a great pleasure to share my work with the scientific community. Today, I'm going to tell you about a story that I've been working on in the past 2 years at Professor David Liu's lab at the Broad Institute on applying the CRISPR technology to construct rewritable multi-event analog recording devices in bacteria and mammalian cells. Recent advances in biotechnologies, including the high-throughput sequencing technology, the proteomic profiling tools, and the metabolomic analyzing technologies, have enabled the detailed mapping of the internal state of a cell, including the sequence of its genome, the set of epigenetic modifications, and proteins and RNAs that are expressed and transcribed. By using these technologies, it is possible for us to take snapshots of a single cell at different time points for detailed analysis. However, these snapshots tell us little about the cell's history and how this history dictates its current and future states. Important cell fate changes are often results of a history of endogenous and exogenous signals. For example, many factors contribute to the process of aging, including telomere erosion, DNA damage, the presence of reactive oxygen species, and ER stresses, and so on. Some of these factors are present in all aging processes all the time, such as telomere erosion, and some of these factors are only present in a subset of aging processes and were there only for a short period of time, such as the presence of reactive oxygen species. By taking a snapshot of the final aged cell, it can be very difficult for us to differentiate the real aging factors that trigger this process and understand how these factors actually affect the aging process. Similarly, during the formation of cancer, the differentiation of stem cells, many signaling pathways are transiently turned on or off in a specific order. It remains challenging for current technologies to record these transient upregulations and downregulations, not to mention their order of function. To record the history of a cell, we decided to build a molecular data recorder that functions in both bacteria and mammalian cells to record critical events in a cell's life, such as environmental insults, developmental decisions, and disease-related behaviors. These molecular data recorder will function like a mini hard drive inside the cell to provide forensic information about cell state dynamics. The Liu lab is not the first lab who is interested in tackling this problem. In fact, constructing durable and minimally perturbative memory has been a long-standing challenge in the synthetic biology field, and it has attracted a lot of interest in the past few decades. The current available synthetic memory devices can be categorized into two categories depending on whether the writing process is transient or permanent. Transient writing often involves control over transcription and translation, and it can be very delicate. On the other hand, permanent writing are often recombinase-based. For these kind of devices, individual states are very stable. However, switching between states is not always as reliable. Plus, orthogonal recombinases are required to record more than one bit of information. Depending on the format in which the memory is stored in, synthetic memory devices can also be categorized into digital memory devices and analog memory devices. For digital computation, it means the device has only two states, on and off. For analog computation, it provides a range of continuous values and in theory can record more information using a single device. Although analog computation has an advantage over digital computation in some setups, there are very few examples available for these kind of devices because analog computation requires a more precise control over complex biological processes. We want to build up a synthetic memory device that can translate exogenous or endogenous stimuli into durable, precise, and predictable markers in the DNA so that the information can be read out even after cell death. Programmable genome editing systems can serve as ideal tools for these purposes, and here we chose the CRISPR technology. CRISPR stands for clustered regularly interspaced short palindromic repeats. It's an adaptive immune system in bacteria to digest virus DNA. The most attractive feature of CRISPR is that it introduces precise double-stranded break in many cell types across species with the help of a guide RNA. To date, 3 major classes of CRISPR systems have been identified, with the type 2 system from Streptococcus pyogenes the most widely used one. With the help of a guide RNA, the Cas9 can unwind the double-stranded DNA and introduce these double-stranded breaks at a designated position in the DNA sequence. These double-stranded breaks can be repaired by non-homologous end joining to introduce random insertions and deletions, or by homology-directed repair to produce precise target DNA replacement if a donor template is provided. So in this work, we developed a system called CRISPR-mediated analog multi-event recording apparatus, in short, CAMERA. CAMERA is a synthetic memory device that will translate exogenous and endogenous changes in stimuli into DNA markers in both bacterial and mammalian cells. 2 systems were developed based on different writing mechanisms using either the Cas9 nuclease or the Cas9-derived base editing proteins. I will talk about CAMERA 1 that uses Cas9 nuclease for bacterial recording first. Remember that we set up the criteria for the recording process a few slides back, that we want the DNA marks created by our device to be durable, precise, and predictable. So with these criteria, we don't want the double-stranded break created by Cas9 to be repaired by the cellular machinery, because in that case, random insertions and deletions will be introduced. so that the change won't be precise. To get around that problem, I designed a pair of recording plasmids called R1 and R2. And I will translate the double-stranded breaks into the ratio shift between these two recording plasmids. Both R1 and R2 are high copy number plasmids with a POC origin and contains the inducible GFP genes. These two plasmids are otherwise identical, except R1 has a premature stop codon in the GFP gene so that it won't be able to produce the mature fluorescent protein. I will co-transform the two recording plasmids at a saturated amount and select for bacteria that contain both plasmids because there should be minimal difference in fitness cost between these two recording plasmids. I expect they can coexist in the same cell for a long period of time. Of these two plasmids, only R1 is the substrate of the Cas9 and guide RNA complex. Once our writing complex is induced by the stimulus of interest, double-stranded break will be introduced into R1 but not R2. Since bacteria are really inefficient in terms of repairing double-stranded breaks, the double-stranded break in R1 will lead to the depletion of these particular plasmid, and the depletion of R1 will initiate the replication of the rest of the plasmids. Remember that these two plasmids share the same origin of replication, which will depict the final copy number of both plasmids in the same cell. So the loss of R1 will lead to the gradual accumulation of R2 so that information can be stored in the form of plasmid ratios. Before I create any recording processes, I want to first verify the stability of the recording plasmid without Cas9. So here I'm showing you two bacteria that contain a different percentage of R1 plasmid from the very beginning. And these two bacteria were cultured for 6 consecutive days with a total dilution number of 10 to the power of 17. You can see that throughout the dilution and culturing period, there is nearly no change in R1 or R2 content in bacteria, which strongly suggests that these two plasmids can be stably maintained in the same bacteria as long as there's minimal difference in fitness cost. Since in this case, we're not inducing the GFP expression yet, so these two plasmids will look almost identical to the E. coli. With the stability of the recording plasmid pair verified, I next started to build the writing plasmid. In writing plasmid 1.0, the expression of Cas9 is controlled by a TET-O promoter, and the guide RNA is constitutively expressed through a LAC promoter. To maximize the differentiation between R1 and R2 by Cas9, these 3 codon changes are located very close to the PAM sequence of the guide RNA so that maximum differentiation can be realized. Here, the CCA is the PAM sequence in the reverse direction. We then PCR'd the GFP gene and the mutated GFP gene from R1 and R2 and incubated these double-stranded DNA with Cas9 and guide RNA in vitro. As you can see from the gel, only the GFP fragment encoded by R1 is a substrate of the writing complex and will be digested by the complex into 2 fragments, but not R2. So now we have the writing plasmid that's doing what we want, depleting one of the recording plasmid, but not the other one. And in this case, the writing complex can be controlled by an antibiotic called anhydrotetracycline, in short, ATC, in this case, with TEO promoter. So the next question we want to ask is whether we can record the presence and the treatment duration of ATC using our writing plasmid 1.0. The bacteria containing a starting ratio of 60% of R1 was selected, and these bacteria were treated with or without ATC. On the right, I showed you the R1 percentage that is analyzed by high-throughput sequencing. So in the absence of ATC, as you can see, the R1 percentage remains stable after 3 hours and 6 hours, while the R1 content responded strongly to the presence of ATC, reduced from 60% to 21% after 3 hours and to 4% after 6 hours. These pieces of data suggest that camera 1 can record the presence and the treatment duration of an exogenous signal in analog form. To enable the system to record more than one stimuli, I further substitute the LAC promoter with a LAC-O promoter in camera 1.1. That is a variant of the previous device I've shown you, the camera 1.0. The LAC-O promoter can be suppressed by LAC-I protein and activated by IPTG. In this case, both ATC and IPTG will be required to initiate the recording process, which mimics the behavior of an AND logic gate. Indeed, we observed the requirement of both small molecules in our recording process. A small imperfection of the AND logic gate was observed when ATC was provided without IPTG. This small imperfection is a result of the likeness of the LAC-O promoter, which has been known for a long time. One of the advantages of our camera system is that it provides analog signals, which in theory can give us more information with a single device compared to binary switches. So to test the analog capability of camera 1.1, I treated bacteria that carrying this device with different concentrations of IPTG ranging from 0 to 150 micromolar at a constant input of ATC. And the R1 content was read out by measuring the EGFP fluorescence of the bacterial culture. As expected, the EGFP fluorescence decreased as the IPTG concentration increased, suggesting that the R1 depletion rate was faster with a higher IPTG concentration. Importantly, the GFP decrease is linear and predictable at low IPTG concentrations, reinforce that we can actually back calculate the IPTG concentration depending on the GFP fluorescence or R1 content we obtain through transient readout or permanent readout. We characterize the R1 content by high-throughput sequencing at the same time and obtained very similar results. So we conclude that both transient readout and permanent readouts are possible with our device. Memory devices can be particularly versatile if it can be reset and re-record. We developed two mechanisms to reset the memory devices. Here I'm showing you one of them. In addition to one guide RNA that targets the unmodified GFP gene, in camera 1.3, we included a second guide RNA unit that's under the control of a Rhamnos responsive promoter. The guide RNA2 will target the mutated GFP gene containing plasmid, in this case R4. And similar to the process that the expression of guide RNA1 that's controlled by IPTG will lead to the depletion of R3. The expression of guide RNA2, in this case controlled by Rhamnos, will introduce double-stranded break in R4, thus reduce the amount of R4 and bringing back the R3 level. Indeed, by repeated treatment of Rhamnos and IPTG in the same cell, we observed multiple rounds of recording and resetting events. In addition, those responsive forward and reverse direction recording can be achieved using different dosage of Rhamnos and IPTG. So from here, I'll switch to the second system that we developed based on base editing. Base editing is a new precise genome editing tool that is developed by the Lab in 2016. Base editors consist of three parts: a catalytically impaired Cas9, a cytidine deaminase, and a uracil glycosylase inhibitor. Together, they will introduce precise GC pair to AT pair mutations in a designated position in the DNA sequence with the help of the guide RNA, without introducing double-stranded breaks. The efficiency of base editing is much higher than homology-directed repair and introduces less insertions, random insertions and deletions. The durable, precise, and predictable single-point mutations produced by base editors can serve as an ideal information carrier for our synthetic memory devices. Because bacteria lack the NIC-directed repair processes, we decided to use the second generation of base editor that contains a catalytically dead Cas9 instead of a nickase that's used in base editor 3 as our protein component of the writing complex. Here I showed you the scheme of how camera 2 functions in bacterial cells. With the help of the guide RNA, BE2 will introduce precise CG pair to TA pair mutations in a designated position in the recording plasmid. If we put the expression of base editor and guide RNA under the control of the stimuli of interest, the presence of the base edited plasmid will faithfully record the presence, amplitude, and treatment duration of the signal. On the left, I showed you a proof of concept experiment that we did with camera 2.0. This is a recording experiment for anhydrous tetracycline again. So in this case, we try to record both the concentration and the treatment duration of this small molecule. As you can see, the editing rate strongly corresponded to the concentration of the small molecule, In other words, the strength of the stimuli. And the total edited plasmid ratio accumulated in a linear fashion with a treatment duration. Importantly, at a low induction level, the editing is happened slowly but constantly. It's easy for us to imagine that with this speed of memorizing, it's easy for camera system to serve as a molecular clock and record hundreds of generations of bacterial cells. So with this experiment, we conclude that camera 2 is a highly responsive analog memory device that can faithfully record the amplitude and the duration of an exogenous signal in the form of single nucleotide change. One of the advantages of using CRISPR technology to generate synthetic memory devices is that multiple signals of interest can be recorded using different guide RNA cassettes that are responsive to these signals. So to test that hypothesis, we constructed camera 2.4, in which we have 3 guide RNA expressing cassette. Each of them is under the control of a different small molecule inducer. In this case, base editor 2 is still under the control of ATC. Here is a test of most of the possible combinations of these small molecule inducers. As you can see here, editing at designated positions can be used to predict the presence of the corresponding writing complex, in other words, the guide RNAs, and hence the stimuli that initiated the expression of these guide RNA, for example, the IPTG one and the arabinose one. And the fidelity of this device is not compromised even when more than 2 stimuli are introduced, reinforce the versatility and multiplexability of our device. Synthetic memory devices that can record event order are of high interest because changes in a cell's environment or endogenous signaling can strongly determine cell fate. With camera 2, we hypothesized that by overlapping 2 base editing positions so that the second base editing event can only happen after the first base editing event happens, we should be able to record a certain a desired order of application. To test this hypothesis, we generated camera 2.5. So here, these 3 G2A mutations that's introduced by guide RNA 5, controlled by arabinose, is located very close to the PAM sequence of the guide RNA 6 that targets by guide RNA 6. And the guide RNA 6 only recognize the 3 A's, but not the G's. In other words, it should not be able to do any editing without the pretreatment of arabinose. So by including another guide RNA target sequence in the modified GFP gene, the device can further record the presence of either arabinose and rhamnose separately. Because in this case, if we only include the second part of the device, the presence of arabinose can be recorded, but the presence of rhamnose, the second signal, won't be able to be recorded without independently, basically. So with this design, we should be able to record these 2 signals independently. So indeed, the editing at position 205 through 207 corresponded well to the presence of arabinose, while the editing at position 129 corresponded well to the presence of rhamnose. And the editing ratio at position 216 versus position 129, both introduced by guide RNA 6, can be used to deduce the actual treatment order. Only the correct treatment order of arabinose first, followed by rhamnose, we can observe a significant editing ratio at these two positions. If the treatment order is reversed or there's no presence of arabinose, this treatment ratio is always less than 0.1. So we conclude that with this staggered guide RNA design, camera 2.5 can record multiple events in an order-dependent manner. Camera can record a variety of environmental signals as long as these environmental signals can be translated into RNA transcription, protein expression, and protein activation. Here I showed you two examples of recording virus attack and light using camera 2.6 and 2.7. One of the very desirable feature of camera 2 is that it can record using a handful of cells. Remember that we record the single nucleotide change into a recording plasmid that is a high copy number plasmid. The plasmid is present in bacteria in hundreds of copies, which means we have hundreds of target sites in each cell, which will greatly reduce the dependence of the recording process on population. Here I use camera 2.7 that is responsive to light as an example to demonstrate these low dependence on population. On the left, I'm showing you the recording in bulk culture that contains 100K cells. As you can see, that the recording is very consistent and the editing accumulated in a linear fashion with treatment duration of light. Importantly, these consistency could be recapitulated with 100 cells and even 10 cells, although larger variations were observed at later stages with less cells. This is the smallest sample size to our knowledge for any kind of analog recording devices. Because base editing, the mechanism also functions in mammalian cells, so we expect what we have developed for camera 2 can be easily translated into human cells. Here we chose established a human safe harbor gene, CCR5, as a recording loci. We also validated 3 guide RNAs that target different positions of the CCR5 gene with minimal crosstalks. By putting the expression of guide RNAs under the control of TET-R or LAC-high suppressed promoters, the presence of doxycycline and IPTG can be recorded in different positions of the CCR5 gene separately and in combination. Strongly suggests that camera 2M is also multiplexable in mammalian cells. So finally, we want to record endogenous signaling event, the Wnt signaling pathway. The Wnt signaling pathway is a major endogenous mammalian signaling pathway. It plays a crucial role in embryonic development processes, and aberrant Wnt signaling is associated with many kinds of diseases. To record the Wnt signaling, we put the TCF, LEF, repeats upstream of the base editor, these will recruit transcriptional factors to initiate the expression of base editor once Wnt signaling is initiated. So we also included another construct that is called TCF-LEF-B3 lock. This construct will express base editor together with firefly luciferase gene so that we can monitor the Wnt signaling both transiently by luciferase luminescence signal and permanently by base editing. We treated the cells with lithium chloride, a small molecule inhibitor of the GSK3 protein. Lithium chloride is known for a long time that is to serve as a Wnt stimuli. So as you can see here, upon lithium chloride treatment, base editing level increased significantly. So as well as luciferase gene, as well as the luciferase signal. Okay, so I hope I have shown you that camera is a faithful and versatile synthetic memory device that can function both in bacteria and in human cells. It can record signal amplitude and durations in an analog format and can be reset and re-record if desired. Multiple signals of interest can be recorded using multiple guide RNA expressing cassette. And camera 2 can also function in human cells to record endogenous signaling, changes in endogenous signaling events. With that, I want to thank Professor David Liu for providing a great environment for doing science and for all his support in this project. Dr. Michael Packer, Johnny Hu, Dr. John Levy, Dr. Fenzhu Xiong, and Dr. Li Wang helped with data processing of this work. And Dr. Ahmed Badran provided one strand that's used for test virus attack in bacteria. And Holly Rees provided the Cas9 protein that I used for in vitro cutting assay. These are the funding agencies that provided provided financial help to this project, NIH, DARPA, HHMI. My postdoc research is supported by Jan Coffin Child's Memorial Fund. I think that's it.
Thank you so much, Dr. Tang. I really enjoyed learning more about CRISPR-mediated analog multi-event recording apparatus systems for cellular events, and I hope everyone else found this webinar educational as well. Okay, so we received some interesting questions, and I would like to answer as many as possible. Let's start with the first one. So, Dr. Tang, what criteria should be considered for the fitness costs to stably maintain recorded plasmid ratios?
Anything that the plasmid put a burden on the cell is a fitness cost. For example, the size of the plasmid, because the larger the size, the plasmid size is, the longer, the more resources it will take for the cell to replicate the plasmid, and what kind of proteins the plasmid expresses. The larger or the more the proteins that the plasmid asks the cell to express, the fitness cost will be larger. In our case, R1 and R2 they have the identical size, and they express the identical set of proteins. So as long as the GFP is not induced. If the GFP is induced, I would assume that R2 will have a slight advantage over R1 because it expresses basically a smaller GFP protein.
Thanks, Dr. Tang. We have another question. To use this camera apparatus, we need to predefine which stimuli we would like to record. Is this correct? And if so, can we use a similar strategy for naturally occurring cellular events? In other words, can we make this recorder universal?
Yes, for the first part of the question, yes. You need to know what you're looking for, basically. The cell is not as smart as humans. It can differentiate, like you give the cell an order, but the cell, it needs to have the pre-inserted device to be able to respond to that order.
Okay, great. Thanks, Dr. Tang. Another question we have is, can camera 1 be applied in human cells as well?
I receive this question a lot. It will be challenging to use camera 1 in human cells because human cells don't really have a very well-defined mechanism of maintaining plasmid copy numbers. So the fundamental assumption we use in camera 1 is that in a single cell, a certain copy number will be maintained, right? So without that mechanism, I would say it will be very challenging.
Okay, we have time for one final question. Moving forward, what will be the next thing for applying camera?
In the near future, our plan is to map cell states using camera, especially in human systems, by tracking multiple signaling pathways of interest. Basically, for example, if we want to know that if a cell is a cancer cell, we can track a few cancer-related signaling pathways, like Wnt is one of them, and maybe NF-kappa B, and OCT, right? So if we can track all these pathways simultaneously in one cell, we should be able to construct, I would say, cell state map with that. That's what I see in the near future.
Okay, once again, thank you, Dr. Tang. In the interest of time, we would like to stop here, and I want to thank all the attendees for joining us today. If you want to watch this webinar again, the recording will be available at genewasweek.com. Also, check out genewasweek.com for more activities, including webinars, exclusive promotions, multiple chances to win $5,000 credit for GENEWAS services, and much more. Thank you.